A-Level Biology 9700, Paper 3, Variant 4, October-November 2025. Question number 1. So, question number 1 is, a plant produces sugars in its leaves. The sugars produced are needed by other parts of the plant and are transported through the phloem as sucrose. Sucrose is transported to the roots where it is stored as starch. Some of the sucrose is transported to the fruit of the plant where it is stored as fructose and glucose. Sucrose is also transported to the seeds of the plant. Water and mineral ions are taken up through the roots where they are transported through the xylem to the rest of the plant. Solutions were made to represent extracts of different tissues and fluids found in a plant. So the different fluid in the phloem, root tissues, seed tissue, fruit tissue, fluid in the xylem, you are provided with 4 solutions: S1, S2, S3, and S4. You will identify the biological molecules present in each of the 4 solutions. Suggest which solution S1, S2, S3, and S4 would represent each of the following extracts. They have given you dilute hydrochloric acid, sodium hydrogen carbonate powder, Benedict solution, iodine solution, and 4 solutions from S1 to S4. So, if any solution comes into contact with your skin, wash off immediately with cold water. It is recommended that you wear suitable eye protection. Now, the question is, you will need to carry out the test for reducing sugar. Test for reducing sugar is also called as Benedict solution, Benedict test. So carry out the test for non-reducing sugar and carry out the test for starch. It means you have to conduct the 3 tests: reducing sugar, non-reducing sugar, and starch. So identify the biological molecules which are present in these 4 solutions. So state the reagent or uh, reagents that are used to test for reducing sugar, and the color or the colors produced if reducing sugars are present. You know that the reagent or reagents they are used is the Benedict solution. This is the reagent which is used to find out the presence of reducing sugars. So the color or colors will be, the color of Benedict solution is blue. So it will change to green. Green color will change into yellow, and yellow will change into orange, and the final color will be the brick red. Now, which color will be produced at that time during your investigation in lab? It depends on the presence of how much reducing sugar is present in the given solution. So these colors will show the presence of reducing sugar in different quantities. Only blue will show the absence of the reducing sugar in the solution. So describe how you will carry out the test for reducing sugar. Very simple. You will take the you will take the S1, S2, S3, and S4 in 4 different test tubes. S1, S2, S3, and S4 in test tubes. Add equal quantity, equal of these solutions, as you can take 2 ml in test tubes and add same quantity of Benedict reagent and heat at least 80 degrees centigrade in water bath. Now we are moving towards the step 1. Label 4 test tubes S1, S2, S3 and S4. Put 1 cubic centimeter of each solution into the appropriate label test tube. Carry out the test for reducing sugar on S1, S2, S3, S4 described in. Record the observed color. So the which color will be present at that time, you will come to know that the reducing sugar is present or absent. If it remains the blue, it means the reducing sugar is absent. If the other color patterns which I have told you will show the presence of reducing sugar. So state the that are used to test for non-reducing sugar and the color or the colors produced if non-reducing sugars are present. So here reagent for is the dilute hydrochloric acid. Uh, dilute HCl plus the other one is sodium hydrogen carbonate. Sodium hydrogen carbonate powder plus Benedict reagent. So these three are used to test the non-reducing sugar. So color will be again the same, that is from blue to green, green to yellow to orange, orange to brick red. So these, any one of these colors except blue will show the presence of non-reducing sugar. Now describe how you will carry out the test for non-reducing sugar. So again, we will take solution in 4 test tubes and then we will add, after adding the solutions into test tubes, add HCl dilute and heat and then and wait for cooling. You know, when we add the acid into the mixture, so what will happen? It will hydrolyze the non-reducing sugars into reducing sugar. So the function of this um, mm, HCl is to hydrolyze the non-reducing sugar into the reducing sugar. So it will break down into uh, glucose or fructose, because the non-reducing sugar which we are going to use in the experiment, that is sucrose, which acts as non-reducing sugar. So after cooling, neutralize it by adding small amount of sodium hydrocarbonate powder until bubbling stop, because it will produce the bubbles which shows the process of neutralization. So when it stops, then you can carry out the Benedict test. As described earlier in the previous part of the question. So it means we have confirmed two tests, that is reducing sugar and not reducing sugar. Now step 5 is label 4 clean test tubes S1, S2, S3 and 4. Put 1 cubic centimeter of each solution into the appropriate label test tube. Carry out the test for non-reducing sugar on described in and record the colors observed. Label 4 clean test tubes S1, S2, S3 and S4. Put 1 solution into appropriate label test tubes. Carry out the test for starch. Record the colors observed in 3. Record your result in an appropriate table. Now here we are going to construct the table. So here you are going to construct the table. And then we will make in this way solutions. You can also make different from it as you can do over there. It depends on you. So this is, we will write down the S1 in this table, S1, S2, S3, S4. So in this column, we will represent color, present or absent. Color, present or absent. Then again color or present, color, color, present or absent. Color present or absent. Now the different test which you will carry out, that is Benedict, non-reducing, starch. So in this way, the color which will be observed, you will write down the color over here. If the color is from green to brick red, then you will uh, take in this way. If the blue color, then you will cross for all these solutions. So in this way, you can find out which of the solution having the Benedict solution and which having the non-reducing sugar. So which one having the starch solution. So you are going to represent at that time when you are conducting your experiment in the lab at the time of examination. Now moving towards the next question, next part of the question, you can see that after making this type of table, you can change the table according to your, but the table, I think it should be a completely representing the data. So complete table 1.2 to the suggest which solutions S1, S2, S3 or S4 could represent each of the plant extracts used. Your results in A3 and the information on plant transport. According to the colors, you can come to know the table which you had drawn in the last question. According to you can write which solution contains. You know the sucrose or non-reducing sugar is present in the phloem, and xylem and it contains water, and you can write down the solutions, which solutions out of 4 solutions contains all these different uh, so you can write there at the time of examination your answer. Now the next is protein could be present in some of the solutions. Describe how you would identify which solution contains proteins. So before going about this fluid in the phloem, I already told you that the sucrose or non-reducing sugar is there, and it should give the uh, positive result for sucrose or in negative for reducing sugar, and starch is also negative, or root tissues contain starch, so it should be positive for starch and reducing and non-reducing for negative. And then seed tissues will also contain starch, so it will be test for iodine. And the fruit tissues contain sucrose and glucose, so reducing sugars will always be there. And fluid mainly water and mineral salt, so it lacks complex organic macromolecules, all these macromolecules. So uh, moving towards the next, describe how you would identify which solutions contain proteins. So for this, the presence of protein, we will conduct the burette test. Burette test. So for this burette test, we will add a few drops of add few drops of burette solution, burette reagent. And burette reagent is made up of dilute sodium hydroxide solution and dilute copper sulfate solution. Uh, B burette solution is made up of dilute hydrogen It's hydroxide. Hydroxide plus dilute copper 2 sulfate, copper sulfate. So the burette reagent is made up of these two solutions. So we will add the few drops of burette reagent and then add the sample solution plus shake thoroughly. Color change from blue to purple will show the positive result. It means for proteins. If color is changed, it can be purple, violet, mauve. So any one of them, the little bit difference in all of these colors, little bit change of the shade. So if the color is changed from blue to purple or mauve or violet, that it shows that proteins are present. Now the next question is there, part B. A scientist investigated the effect of light on the concentration of sugars in a plant for 24 hours. The plant was kept in the dark for the first 8 hours and then exposed to light for the remaining 16 hours. Samples were taken from the leaf and from the phloem sieve tubes. The concentration of sugar in each sample was measured. So the results are shown in table 1.3. Uh, and plot a line graph of the data in table 1.3 on the grid in figure 1.1. Use a sharp pencil. Now we have to draw the graph. So for this purpose, we need this data from the table. So with the help of this, we can find out, or we can draw the graph. I need these values to draw the graph. So the values in the table has been given. That is the independent and dependent variable. We have to choose which one is the independent variable. So the independent variable in this table is time per hours, which will be taken on the x-axis, and other is this is independent variable. So it will be taken at x-axis, and these two will are dependent variables which will be taken on y-axis. So the value starts from time starts from 0 and the values are differently 0.33 less than 1. So we have to take all these two curves will be formed in this. So moving towards the formation of this graph. So wisely you have to select the scale over here. As the scale which you will be select on x-axis, the on x-axis the time or the in hours. So in this way, time per hour. On y-axis, you will choose the dependent variables, which are concentration of sugar and others. So concentration of sugars per micromole. So the it will be, uh, the different time which has been on x-axis. It will be from 0 to 25 hours. 0 to 25 hours. So we will do this scale as two small squares will be equal to 1 on x-axis, and on y-axis 10 small squares are equal to 0.1. Small squares are equal to 1, 0.1, 0.1. So in this way we will choose the over here. So students, it's very difficult for me when I'm drawing the graph on my laptop screen. So you have just note down the points which I am doing over here. So you can draw your graph by yourself very easily. I know, I have trust on you students. So I have to select from, I can do it 0. The value which will be start right from here is 0, and then 1 2 3 4 5 6 7 8 9 10. About 10 is 5, 0 5, 10, 15. Very difficult for me to draw, so be careful when you are drawing on your in your examination. 15, then it will be 20, and here it will be 25. So on the other side, once it is 0.1, it is 0.2, it is 0.3, and it is 0.4. You can count down these squares because I'm just giving you the hints that in this way you can draw. I'm not properly drawing, I'm not properly measuring anything, just giving you the idea. Okay, so the first which we are going to draw is with the values which I will take for this is for leaves. The first which we will leave for the leaves, the first value which is at 0 is 0.38. 0.38. So it will be over here. We will make a small cross. It is not small cross because I'm reminding you again and again that I'm working on my laptop. The second is the value is at 5 hours 0.21. 0.21. So it will be very close to this one. And the third value is 0.13. So it is at 8. So it will 13. It is at 8. So it is very close to this one. And the fourth one is which is at 0.24 at 15 hours. 15 hours 0.24. It will be 0.24 about this. and the next one is at 0.39 at 24. So 24 is 0.3. So we will draw these lines. So the curve will be of, for this is the for leaves. The next is for phloem sieve tubes. The first value, which is at 0.22, it is here, and this 0.17 at 5, 0.17 at 5 will be round over here, and 0.11 at 8 hours, 0.11 and 8 hours, and the other one at 15 is 0.16, 0. This is 1, and this is 0. More than the center. And at 24, it is 0.22. 0. Now we will draw these lines. And this one is for leaves. And the lower one shows the phloem sieve tubes. So I have given you the rough idea how you can draw this graph with two curves. Here from these two graphs, it has been, you can take a better idea that that the values, concentration of sugar is always higher in the leaves. You can see the curve, it is above for leaves, and the lower one is phloem sieve tube. So it will also show the lower concentration of sugar. Describe the trends for the concentration of sugar in the leaves and phloem sieve tube shown in figure. I have told you this idea. The sugar decreases from 0 to 8 hours. In both curves. Both curves are the same, but they are little bit more concentration and the other one has lower concentration. Then from 8 to 24 hours, the sugar concentration increases. In both locations. Now suggest an explanation for the trend in the data when the plant was in the dark and when the plant was in the light. So explanation: for plant in the dark, photosynthesis takes place, so no sugars are produced. So no sugars are produced. Plant use up the existing sugars for respiration. And what about 8 to 24 hours? The plant undergoes photosynthesis. Undergoes photosynthesis in the which rapidly produces triospores, sugars are producing. So next question is the percentage increase. Calculate the percentage increase in the concentration sugars in the leaf between 15 to 20 hours. So here you have to show your working. From the table, we can see the values as move towards the table. 15 to 20 hours, the value was 0.24 and it was increased 0.39. So the initial value is 0.24 and the final value is 0.39. Value is 0.24. Final value is 0.39. So the formula of percentage increase is initial value minus final value. Very bad handwriting. Initial value minus final value divided by initial value multiplied by 100. So the initial value was 0.24 minus 0.39 divided by 0.25 multiplied by 100. So to calculate this value, minus 0.24, 0.15 multiplied by 100. Divided by 0. So it is equal to 62.5%. You can calculate it by yourself. So it was all about the question number 1 for your examination paper 3-4. Please like, share, and subscribe my channel to watch more videos. And thanks for watching this video.